The film tells us about the mode of action of innovative drug product Neovasculgen, which was developed by the Russian biotechnological company Human Stem Cells Institute. Neovasculgen is the first drug product in the world which is intended for treatment of chronic and critical limb ischemia, with mode of action therapeutic angiogenesis. The mode of action of Neovasculgen introduces a new approach to treatment of ischemia. The mode of action of Neovasculgen introduces a new approach to treatment of ischemia, use of an evolutionary programmed process of formation and growth of blood vessels. The product is a ring DNA molecule plasmid. The plasmid penetrates to a cell via endocytosis. Cell membrane forms niches, emboles, which are then transformed to specific follicles, vacuoles. Such vacuoles get plasmid within a cell. Nuclear membrane, which consists of two phospholipid layers, as a cell membrane contains a large amount of nuclear pore complexes of various diameter. Ions, proteins, RNA and ribonucleoproteins are transported through pores, both from nucleus to cytoplasm and from cytoplasm to nucleus. Small molecules freely diffuse through nuclear pores, but large molecules are transported with consumption of ATP energy. The transport may be increased if a transported molecule carries the so-called signal of nuclear localization. In a nucleus, information or matrix RNA is formed with one of plasmid chains. The part of DNA only is copied which contains information about necessary Vgf protein. A specific enzyme, RNA polymerase, is required for transcription. Moving over a DNA chain along the necessary gene, RNA polymerase selects nucleotides by complementary principle and combines them to a chain of mRNA molecule. At the end of the gene, the enzyme receives a signal as a definite sequence of nucleotides, which means end of transcription. Mature mRNA goes off from DNA. Immature pre-mRNA converts to a mature mRNA and goes to site of protein synthesis. Translation is a protein synthesis from amino acids based on information or matrix RNA. For the process, cells of all organisms have specific organelles, ribosomes. Ribosomes are ribonucleoprotein complexes formed from two subunits, large and small one. The function of ribosomes consists in recognition of three-letter mRNA codons, correlation with appropriate anti-codons, tRNA carrying amino acids, and the consolidation of such amino acids with growing protein chain. Moving along mRNA molecule, the ribosome synthesizes protein in accordance with the information inserted to the mRNA molecule. To recognize amino acids, the cell has special adapters, molecules to transport RNA. Such cloverleaf molecules have anti-codon region complementary to mRNA codon, as well as another region to which amino acid relevant to the codon is attached. Subsequently, post-translation changes of Vgf protein occur. Secondary, tertiary, and quaternary protein structures are formed with the enzyme involvement and energy consumption. Recovery of Vgf protein from cell occurs with exocytosis. The process is exactly the opposite of plasmid penetration to a cell. A Vgf macule migrates to a cell membrane, merges with it, and Vgf goes outside the cell. Getting into ischemic tissues, Vgf protein binds with Vgf-sensitive receptors on endothelial cells in pre-existing vessels. It results in activation of endothelial cells due to changes of Vgf receptor configuration. Activated endothelial cells begin expressing proteolytic enzymes which in their turn dissolve basal membranes surrounding source vessels. Subsequently, under effect of matrix metalloproteinases, intercellular matrix is dissolved. So space occurs for migrating endothelial cells. When mechanical barriers are eliminated, proliferating endothelial cells begin freely migrating to ischemic zone. Migrated endothelial cells bind with each other and form tubular structures. Tubules grow, elongate, and begin forming vessel loops. Formed vessel loops differentiate to arterial and venous vessels. Subsequently, new blood vessels get mature parientally, consolidating with other cell types, smooth muscle mural cells. Vascular architecture is stabilized, blood flow begins in newly formed mature vessels. The clinical studies have shown efficacy of the product. In accordance with the study data, the walking distance reliably increased up to 173% in 90 days after the drug administration and in a year in 3.5 times.